talk about the functions of DNA. So in our bodies, DNA must do three important things. It must carry information from one generation to the next, from your grandparents to your parents to you to your kids. It must put that information to work by determining the heritable characteristics of organisms. And it must be easily copied so that when a cell goes through mitosis, each new cell will have a complete copy of the DNA. So DNA structure is that DNA is a very long molecule, and it's made up of single units or monomers that are called nucleotides. And there are three individual parts to a nucleotide. There's the 5-carbon sugar called deoxyribose. There's the phosphate group. And there's one of 4 possible nitrogen bases, which are adenine, thymine, guanine, and cytosine. So again, our nitrogen bases are adenine, which we symbolize by A, guanine symbolized by G, cytosine symbolized by C, and thymine symbolized by T. We consider adenine and guanine to be purines. It just has to do with their shape. And the way that I like to remember that is that adenine and guanine are pure as gold, whereas cytosine and thymine are considered pyrimidines. And I like to remember them because both cytosine and thymine have Y's in them, just like the term pyrimidine. So an easy way to remember those. All these things come together to make the structure of DNA. The backbone of DNA, or if we were to look at DNA like a ladder, the sides of the ladder are going to be made of your sugar, which is deoxyribose, and your phosphate group. And then the bases stick out sideways from the backbone. These are the parts of the ladder that you would step on. And remember that A always pairs with T and C always pairs with G. Always, always, always. The only reason that would not happen is if there was a mutation in your body, which we'll talk about in the next unit. The nucleotides can be joined together in any order, any possible sequence, and the sequence that they are joined in is what determines your genes, which code for proteins, and the proteins help you express those genes, like your hair color, your eye color, whether you have a widow's peak or not, and things like that. And finally, all those things together create what is called a double helix shape. So it's if you took a ladder and you just twisted it, you would get the double helix, which is the shape of DNA, as given to us by Watson and Crick. All right, so in order to really get a look at what DNA looks like, I decided that I was going to take you from big picture to little picture and kind of show you how small DNA really is and what it looks like when you get up close. So here I have a person, and I just took a snapshot of her thigh. And if I were to use a microscope and get really, really close, I might see something like this. All of her thigh muscles, the circles are the nuclei. And I'm still not small enough to be able to see DNA yet. What I need to do is I need to get even closer. So I took a snapshot of just one of those thigh cells. And as you can see, I have all of the most important structures in there. The nucleus and the nucleolus, some mitochondria, vesicles, my endoplasmic reticulum, my Golgi, which are useful for protein synthesis. But still not small enough because we still can't see the DNA yet. So DNA, we know from talking about it before, is housed in the nucleus. So I took a bigger picture of just my nucleus where I could see the nucleolus, which is this orange dot right here, and I could see a closer look at my chromosomes. Remember that in human somatic cells or body cells, there are 46 chromosomes. If we're talking about human gametes, either eggs or sperm, we're going to see 23 chromosomes. But anyways, looking at one of those chromosomes closer up, we're going to see this double helix twisted ladder shape where the sides are going to be made of my sugars, deoxyribose, my phosphate groups, and the orange in the middle is going to be my nitrogen bases. Still not small enough yet because we really want to see a picture of what these deoxyribose and these phosphate groups and these nitrogen bases look like. So I took an even bigger snapshot of that section of DNA right there, and this is what I get. On my sides of my DNA over here, it's the green part, I get alternating phosphate groups and sugars. I drew them with circles and triangles so that we could see that they alternate back and forth. All right. In the middle, if I'm thinking of this as a ladder, the parts that you would step up, the rungs of the ladder, those are made by my nitrogen bases, my adenine, thymine, cytosine, and guanine. Remember, adenine and guanine are my purines. They have a double shape when you look at them. And if you look at cytosine and thymine, those are my pyrimidines. They have a single shape when I look at them. But always, 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 A and T pair together and C and G pair together. They are held together in the middle by something called hydrogen bonds. Hydrogen bonds are really important because they are weak bonds. They are just strong enough to make sure DNA doesn't crumble apart, but they're strong enough so that they're weak enough, I should say, so that when it comes time for mitosis or meiosis and we have to replicate the DNA, we can really easily break these bonds apart so that we can get around DNA replication. The last thing I did here was I sort of pulled out a gene for you, and I showed you the way my base pairs line up, my A's with my T's and my C's with my G's always. Now, in human beings, let's say this is a gene for hair color. Every single person that has brown hair is going to have a pretty similar set of letters, set of base pairs in order. If I am somebody with blonde hair, I might have a couple of these base pairs changed out for some different letters, and that would still be different for somebody with red hair or black hair. So my genes will have different orders depending on what they code for, which we'll talk about when we get to protein synthesis and genetics.